Hello, Professor. I'm Kristen Kelton, and it's nice to meet you. Reading technical information, then synthesizing it, and presenting it in a way that one's audience can understand it is a necessary skill in both science and life. As I explain the merits of an article, it will be as a demonstration to my students on how to read and interpret that technical information. Moreover, there will be assignments embedded throughout the video so that you could see how I would teach this concept. So from here on out, I'm going to be acting as if I'm teaching this to my students in a flipped classroom, which is both in class and online. Hello, class. We're going to take a little departure from the normal chemistry we're currently learning and just stop for a moment and learn to read, interpret, summarize, and communicate technical information. That's our primary objective. See, it's important that you can read, interpret, and accurately communicate technical information. Recall that after a scientist does research, he must share that information through both spoken word at conferences and through written publications. Moreover, scientists are always reading other publications, so it's vital that you can read, interpret, and write about technical information, and then be able to explain it to a layperson in a way that makes sense. Along the way, we're going to practice small snippets of reading and summarizing technical information. Your final assignment for this section is to read an article and write a summary that conveys the information in a way other chemistry students can understand. Don't forget, though, at the end of the semester, you have a research project in which you will apply these skills. We'll discuss that at the end of this lesson as well. So to get you there, we're going to explore an article together where you will learn how to read, interpret, and communicate technical information. Throughout this presentation, you will be asked to pause the video while you perform small tasks and assignments. Ultimately, though, this article fits into our course under current research on environmental detection devices. So again, our primary objective is to focus on reading, interpreting, and summarizing technical information. Our secondary objective is applicable to what we're currently studying, which is how are microfluidics used in detectors, and why are those detectors so important? Before we jump into reading and writing and interpreting technical information, let's review what it is we've been studying, and that is quantitative analysis in detecting heavy metal pollution. Now, we know that many heavy metals exist in the environment, including mercury, copper, zinc, cadmium, lead, and chromium. While they are naturally in the environment, they often enter the environment via industry and medicine. Mercury, for example, is used in electrical switches, thermometers, barometers, dental fillings, light bulbs, paints. Ethyl mercury is a preservative in some vaccines. Mercury in particular is toxic to both humans and wildlife. In both humans and wildlife, mercury can cause kidney and liver damage. It can disrupt the nerves. It can cause issues with the cardiovascular system, the gastrointestinal system. And mercury is very dangerous to pregnant women and fetuses because it can cross into the fetus. Mercury poisoning often mimics other issues, so it's difficult to diagnose. It can also affect wildlife through reproductive issues, growth issues, very similar to that which occurs in humans. If scientists can detect ultra trace amounts of heavy metals like mercury, then they can locate the source of pollution. By doing that, they can then give that information to government officials and different agencies, which can then guide policy and rectify the situation, and or even educate the public on necessary precautions. Before we go any further, I want you to read a bit about mercury in the environment, its toxic effects, and how one set of researchers are trying to find a new and unique way to detect it. I want you to read this article, which can be found in Canvas, and then I want you to go ahead and do the DQ assignment you will find in Canvas. Write a DQ response that covers how mercury enters the environment, the current methods of detecting mercury, the purpose of this study, which includes how they are going about identifying and detecting mercury, And lastly, what conclusions these researchers came to. And don't forget, you need to respond to at least 2 other DQs by 2 different students. Let's take a moment and discuss sample preparation and detection of mercury in the environment. Much of this you should have gotten out of the article you just read. First, they must prepare the sample, and this can be done through a variety of techniques. Just to throw a few at you, there's several different micro extractions, including single drop, hollow fiber, liquid phase, dispersive liquid, and solidified floating organic drop. Now, once the sample's been prepared, they can go ahead and analyze that sample. Now, 5% of the time, scientists use gas and high-performance liquid chromatography. 14% of the time, they will use electrochemical instruments, including the voltammeter and potentiometer. But 81% of the time, they use spectroscopy or spectrometry. Now, there's lots of different techniques here. A few include atomic fluorescent spectrometry, atomic absorption spectrometry, several different kinds of inductively coupled plasma emission spectrometry, spectrophotometry. They also use cold vapor atomic absorption and cold vapor atomic fluorescent spectrometry. The type chosen really honestly depends upon the sample and its matrix. Is the sample aqueous, solid, gas? What matrix is it in? A couple of newer techniques include stable isotope analysis, and this is where Zoe et al. Focused on analyzing stable isotopes in the ocean. They use something called a multi-collector inductively coupled plasma mass spectrometer that allowed them to trace sources of mercury in the oceans, how it enters the ocean, how it cycles through the ocean. They did have to pre-concentrate their sample due to the low concentrations of mercury, and they had several different ways they went about doing that: ion exchange resin, purge and trap, co-precipitation. There are lots of advantages and disadvantages to those different techniques. I'm not going to discuss them here, but feel free to research them. It's pretty interesting. One other new model is called the into-fish model, and this was to quantify mercury at all stages of the fish cycle. And this was to focus on bioaccumulation because this is very dangerous to humans and the animals that eat fish. They were able to look at both inorganic and methylmercury in sediments, seawater, and living organisms. They tracked the pathway of mercury from sediments and seawater into fish and marine life. It allowed them to predict how mercury pollution in a particular site could affect the entire biogeochemical system. Before I jump into this article I promised I would share with you, I want you to go ahead and read it and do a little bit of an assignment. Now, this is not going to be graded because our focus in this class is on learning to learn. By reading and annotating this article, you can compare how you interpreted it to how I interpreted it. Now, I'm the teacher, but I am not perfect. It's entirely possible I did not interpret this correctly. In fact, in class, we're going to have a discussion that compares my analysis of the article to yours. So as you listen to my presentation, annotate your notes, pointing out where we differ, where we agree. Also, write questions that we can clarify later. The focus of this is to learn to read and interpret difficult technical writing. And if you're unsure of words or procedures, do what I did. Look them up on Google, read some other sources, watch some videos. Now, what I do want you to write about in your own words is to summarize their purpose, how they built their detector, what their conclusions were, and how this helps society. Now, again, you do not need to publish this. This is not a graded assignment, but I do want to have this discussion in class with you. Now that you've read the article, let's discuss their purpose, which was to develop a miniaturized detector combining microfluidics and electrochemistry that could detect mercury and copper simultaneously in various matrices, including blood and water. They wanted a device that was small, versatile, non-toxic, easy to use, and inexpensive, and something that could be used outside of the laboratory, something they call point of care. Take a moment to look at what you thought the purpose was. Write a note as to how close your assessment of the purpose was to my assessment. We'll be discussing it in class. The first thing Mohan et al. Did was make the detector. Remember, they wanted to use microfluidics, which just means etching microchannels on glass that can direct and move water samples. They chose 5 centimeter squared glass indium tin oxide slides because they were transparent, versatile, and conducted electricity. They used a carbon dioxide laser to etch the microchannels into the glass to form the 3 electrode channels. Now this is where it gets interesting. They wanted to create a very small device. They used 20 millimeter long microchannels, but they needed to determine how wide the channels needed to be and then how big the gaps were in between the channels. They needed to do some experimenting on this, and when they tested for the gaps, as you can see in this graph, they tried several different sizes. They did gaps of 0.5 millimeters all the way up to 1.5. They used a standard redox mediator and they tested it using cyclic voltammetry to measure the current. But when they did this, they discovered that the 1 millimeter gap had the best peak current. And you can see that by the red line in the graph. Then they tested the width of the microchannels. Again, they wanted to make it as small as possible, and they really wanted to use 0.1 millimeter widths, but the machine they were using just was unable to make channels that small. And even at 0.3 millimeters, only partial channels formed. And as you can see in that picture, it's kind of hard, but if you go back to the article and look at the picture, you can see it a little bit more clearly. The smallest width the machine could make that allowed for good electron transfer rate was 0.5 millimeters. So their dimensions were 20 millimeters by 0.5 millimeter wide with 1 millimeter gaps because this led to the best electron transfer rate. Before we get into the 3 electrodes that they used in their machine, I want you to go do a very quick assignment. There's a link in Canvas and on the PowerPoint to an article and a video on potentiostat and electrodes and what they do. So pause the video, go take a look at that, and then come back. All right, now that you're back with me, we know that a potentiostat, which is what these guys were creating, regulates voltage while measuring current. Typically, the current is measured and then correlated to something like concentration of the analyte, which is what Mohan et al. Did. Their potentiostat has 3 electrodes. The reference they used was made from silver chloride ink. And then, of course, there were the working and the counter electrodes. The working electrode is where the chemistry actually occurs. The reference electrode's job is to apply constant potential without current, and the current flows from the counter electrode. Well, so far their device was open, and they needed to enclose it so they can include a pump to direct the water flow. They used soft lithography to create the channels on a mold and then poured polydimethyl siloxane, which is a synthetic polymer kind of like rubber, into this mold and let it cure overnight. They then bonded that layer to their plate, their glass chip, to enclose the whole system. They were then able to add a pump that could control the water flow, and lastly, they attached the electrodes to a smartphone and app to monitor the data. Once they built the device, it needed to be tested. The first thing they did was test the flow rate to adjust for the maximum flow that gave them the best electron transfer that separated mercury and copper into separate peaks. They concluded that the best flow was 90 microliters per minute, as you can see from that peak on the graph. From this, they then performed a scan rate analysis for both mercury and copper. This allowed them to calculate the diffusion coefficients for both metals. The slope from those 2 graphs were used to do some math to find the diffusion coefficients. Once they determined flow rate, they needed to know if they could simultaneously measure 2 different ions in a sample that often coexist together, mercury and copper. They used square wave voltammetry. Well, not only did copper and mercury have separate oxidation peaks, as you can see in this graph, but they were able to show that they could detect them simultaneously. They then used the data and linear regression models to find the slope and regression coefficients for both metals. This led to calculations for the limit of detection, which was 2.625 micromolar for copper and 9.248 micromolar for mercury. They then tested interference with chromium, cadmium, zinc, and lead and found that they did not interfere even though they were in the matrix. Once the device was created and tested with laboratory chemicals, they went ahead and tested their detector on tap water, lake water, and blood samples. Now, the samples needed to be prepared. The soil was filtered from the water and blood samples were centrifuged. All the samples were then spiked with 3 known concentrations. By spiking the solutions, the researchers knew how much they added in. When they tested it with the square wave voltammetry, they knew they should get their concentration back or pretty close to 100%. If not, then they knew something was wrong with the matrix that was interfering with their results. The researchers again used square wave voltammetry and the results to find the recovery percentages. This standard addition approach resulted in high percentage recoveries for mercury and copper in all 3 samples. So they knew that their device was working and giving them valid results. Besides just being able to create this detector, why did Mohan et al. Set out to do it? Well, being able to test groundwater, tap water, bodily fluids like saliva, blood, urine, and sweat for heavy metals is truly important. We know mercury is toxic to humans and wildlife, and while copper is necessary for cells, too much or too little is dangerous. Being able to detect these heavy metals will help officials locate sources of heavy metal pollution and educate the public. Heavy metal detection is valuable in medical diagnoses and treatments. While we do have many instruments and techniques to analyze heavy metals, these instruments are large, expensive, and not practical in the field. Mohan et al. Set out to create a miniaturized and accurate microfluidic device that could be used in what they call point of care, that is, not in a laboratory, but out in the field. The device, as they shown, can detect multiple ions simultaneously, and with some minor modifications, they do believe their device could be used to also detect cadmium, chromium, lead, and zinc. To wrap up this little mini unit on communicating and summarizing technical information, here's your assignment. Go into Canvas and read this article, and then write a 250 to 500 word summary. I want you to include the purpose of this experiment, in detail what was their quantitative analysis technique. Please emphasize what was unique about this technique. What were the results? What were their conclusions? And lastly, how will this research benefit society? For more information on the assignment and to see the rubric, please look at Canvas. I have one last little assignment for you. Keep in mind that at the end of the semester, you will conduct an experiment of your choosing, and you will then write a paper that includes an abstract, literature review, methods, results, and conclusions. I want you to see how this mini lesson on communicating and understanding technical information will be applicable to you later in this class as well as other classes down the road. So to start you thinking about this big project, go back through previous labs or for ideas, or just think about something that is interesting or important to you. On Canvas, submit 3 possible research ideas. No, you're not locked into these. For each research idea, what would be the purpose of your experiment? What kind of data would you collect? What kind of equipment or procedures would you need to employ? And ultimately, what would be important about this research? Why is it applicable to society? I promise we're almost done with this video. I want to review the objectives with you one last time. The purpose of this mini lesson was to review reading, interpreting, summarizing, and communicating technical information. By now, you should be able to explain how Mohan et al. Created their device, what it was used for and how it worked, and the importance of having such tools. What I need to know from you in class, or please email me or contact me through Canvas Messenger, what further support do you need to prepare yourself for your final end of the semester project?